The method of giant patches combined with ion-selective microelectrodes is a versatile method used to detect and quantify ion fluxes. Specifically, it is used to measure the proton fluxes generated by the activity of the electro-neutral ion transporter, the sodium-hydrogen exchanger, or NHE, which is expressed on the cell membrane. The sodium-hydrogen exchanger generates a gradient of protons in close proximity to the plasma membrane, indicated here as an orange ring. This gradient of protons across the cell membrane is measured by whole cell patch clamp recording combined with a technique that uses a proton-selective microelectrode. The proton-selective microelectrode is moved close to the cell surface where it records the flux of protons moving out from the cells, shown here in position A, and away from the cell surface where it records the free proton concentration present in the solution, indicated by position B, and then back to position A, close to the cell membrane, in a repetitive oscillatory movement. The voltage difference from A to B is recorded and is dependent on the representative of the NHE activity.
Transport of ions such as proton across the cell membrane affect processes essential to life. Therefore, changes in pH are tightly regulated by an array of molecules, including the sodium proton exchanger, a plasma membrane proton extrusion system. The oscillate pH microelectrode combined with all-cell patch clamping is a novel approach to study the proton flux mediated by the sodium proton exchange activity. Application of this method